Bismillahirrahmanirrahim. Today, inshallah, I will talk about cellular respiration. And as we have talked about this previously in our summary video, this is when a cell retrieves the energy from the glucose. So plants, autotrophs make energy. They store energy, if they wish to, in the form of glucose. And however, that's a stored form of energy. And this energy needs to be unstored or retrieved into usable forms such as ATP. And this process is called cellular respiration. So in the summary, the process of photosynthesis and cellular respiration therefore are linked in a way. Chloroplast in plant cells, with the aid of sunlight, they capture the sunlight and with the aid of chlorophyll molecules, and they produce glucose and oxygen as byproducts. So glucose is the stored form of energy produced by the chloroplast, and a byproduct of photosynthesis is oxygen. Now, the process of retrieving that energy occurs primarily in the mitochondria, and this process is cellular respiration. So mitochondria takes glucose and the oxygen, which was a byproduct of photosynthesis. It uses the oxygen to unlock the energy, so to speak, as we have talked about before. And this process occurs primarily in the mitochondria. As a result, the process produces a lot of ATP, the usable form of energy. One glucose molecule, approximately when the process functions at its best, produces about 36 to 38 ATPs from one glucose molecule. The byproducts of this is carbon dioxide and water. Since there are 6 carbons in the one glucose molecule, 6 molecules of carbon dioxide are produced. And as you can see, these byproducts of cellular respiration, carbon dioxide and water, are precursors, again, to photosynthesis. Photosynthesis is a process where plants take ambient carbon dioxide and water and use that to capture the light from the sun. So these two processes, photosynthesis and cellular respiration, are the great cycle of nature, so to speak. One generates the energy in stored form, the other one restores the energy, or releases the energy in the form of ATP. So here's a mitochondria. Not all cells have chloroplast, of course. The cells that are able to do photosynthesis have chloroplast. But all cells, all eukaryotic cells, have mitochondria because they all need to unlock the energy that's in sugars, such as glucose. Mitochondria is an organelle, and it's a two-membrane organelle. So it has an outer membrane and an inner membrane. So there's an outer membrane and an inner membrane. And in between these two, there's a space, intermembrane space. That's the intermembrane space. The outer membrane, the inner membrane, and the intermembrane space that's over here, and that's actually very important. So a mitochondria has this convoluted inner membrane, and if you wanted to draw kind of like a mitochondria, you kind of draw it like this. And if you wanted to draw a chloroplast, on the other hand, you would draw like those sacs of thylakoids like this and then granum like that, right? So cartoon drawings of mitochondria would be this, and this would be a chloroplast. This is an electron micrograph of a mitochondria, and you can see the outer membrane and the very convoluted inner membrane. Now these valleys and these hills over here, so to speak, these ridges, they're called cristae. That's plural. The singular one is crista. So a bunch of cristas give cristae. Mitochondria are interesting in that they have their own ribosomes, mitochondrial ribosomes. They in fact have their own DNA as well. It's like a circular DNA, just like bacteria. So almost they're like mitochondria are like almost like baby bacterias, so to speak. And this, of course, is where the release of energy occurs. Same picture basically suggests again the outer mitochondrial membrane and the inner mitochondrial membrane. Here again shows the DNA, which should really be actually circular DNA like this, stranded. So it's a circular DNA, little baby strands like that. And inner membrane and outer membrane, intermembrane space again, and those are the cristae. Now the inside of the inner membrane, this space, is given a name, and that's called the matrix. It's called the mitochondrial matrix. Mitochondrial matrix. So the process of cellular respiration, again, is taking glucose and using oxygen to unlock the energy in the glucose. That's why we breathe oxygen. So when we take a breath, actually, the oxygen that we use is really predominantly for this process. And we can't store oxygen in our body because it's a very small molecule that diffuses out. So we breathe it in. So glucose plus 6 oxygen molecules releases 6 carbon dioxide molecules because there's 6 carbons in the glucose and 6 waters and lots of energy. So this is the overall equation for the process of cellular respiration. Now, just like photosynthesis, you can write a nice overall equation, but this process itself is quite complicated and occurs in different stages. The same is true for cellular respiration, as we have seen before. The first part of this 4 stages of the process is called glycolysis. Now, glycolysis is taking the glucose molecule and changing into 2 pyruvate molecules. Okay, so glycolysis is a process of taking glucose and making pyruvate molecules. And this process occurs outside the mitochondria. This occurs in the cytosol. Okay, and this generates ATP. It generates the net 2 ATPs. And it also generates NADH plus H plus, or sometimes it's written as NADH. Now this NADH is going to go inside the mitochondria into the electron transport chain, and which is the thrust of all of these reactions. It generates these electron taxis, the electron carriers, and which they take the electrons to the electron transport chain, which generates lots of ATP. The pyruvate then travels inside the mitochondria, and the pyruvate is converted into acetyl-CoA. So this is the second step, so pyruvate conversion. And the acetyl-CoA then joins and participates in the Kreb cycle, or the citric acid cycle. And this generates a lot of ATP, okay, and also generates electron carriers, NADH, and FADH2. This is FADH2. They both have 2 hydrogens, because I said over here, sometimes you just don't write the 2 hydrogens just to make it simpler to write. And though these are electron carriers, they also go to the electron transport chain, and you generate lots of ATPs over there, like 30 ATPs or so. Okay, so the majority of energy that's generated is by this electron transport chain, which is actually in the inner membrane at the mitochondria. So this reaction occurs in the cytosol. This process occurs in the cytosol. The remainder of the processes occur in the mitochondria. This in the matrix of the mitochondria, the Kreb cycle in the matrix of the mitochondria, and electron transport chain in the membrane, the inner mitochondrial membrane, as we've seen. This is the same picture, again, for emphasis. So here's glucose coming from a blood vessel, and it goes into the interstitial tissue outside of the cell, and there are carriers for glucose which take the glucose in. The glucose then is broken down into pyruvate in the cytosol, this first process called glycolysis, which gives us 2 ATP. The pyruvate then goes inside the mitochondria. It is then converted, as I said, to acetyl-CoA, then which participates in citric acid cycle or the Kreb cycle. All of these processes, the main goal seems here, as you can see, is to generate electron carriers, is to generate high energy electrons, to capture those high energy electrons and donate them to the electron transport chain, which generates lots of ATPs. Here it says 32 ATPs. Sometimes you see 32 ATPs, 30 ATP. The variation is because the electron transport chain sometimes gives 30, sometimes gives 32 ATPs. Okay, so this is the big picture of the process. So we're going to talk about each of these 4 steps individually. So the first part of the cellular respiration is, of course, glycolysis. Glycolysis is breaking down of glucose, as we have said before, into 2 molecules of pyruvate. Glucose is a 6-carbon molecule, and pyruvate is a 3-carbon molecule, as you can see over here. 1, 2, 3. Now, pyruvate, not to be confused with pyrates, pyruvate has a lot of cash, so to speak, and that's pyruvate is going to go into the mitochondria for the remainder of the processes. Now, this process, glycolysis, going from glucose to 2 molecules of pyruvate, gives 2 NADH, which will go into and donate the electrons eventually to the electron transport chain. This process generates a net of 2 ATPs. In other words, the first part of the process actually requires 2 ATPs, and then it generates 4 ATPs. So there's a net of 2 ATPs that are generated. So the initial part of glycolysis, the first 5 reactions, actually require energy. And then the second half of the glycolysis generates energy. So 4 ATPs are generated, but it's a net of 2 ATPs. Now, so here's, again, glycolysis. Now, the remainder of these processes, where the pyruvate goes into the mitochondria, is dependent on the presence of oxygen. Okay? As I said, we're dependent on our breathing on a minute-to-minute basis. Anytime this process is truncated, then literally we cannot survive, so to speak, right? However, there are organisms that are called anaerobic organisms that do not require oxygen. How is that possible? How do they generate the energy? Well, they generate their energy solely on the basis of glycolysis. Okay, and this process, the NADH that's generated during this process, is not going to the electron transport chain because they can't do it, because that process is dependent on oxygen. So all of this process, the electron transport chain, is an oxygen-dependent process, as we'll see. If that's defunct, if that's not available, then we have anaerobic respiration. Then you regenerate NAD from here using fermentation process, making either ethanol or lactate. So pyruvate has 2 destinies. If oxygen is present, it will go into the mitochondria. If oxygen is absent, it will be metabolized, and ethanol or lactate will be produced to regenerate those NAD. We will talk about that again, show something. So the first stage was glycolysis. The second stage is conversion of pyruvate to acetyl-CoA. And the third stage is oxygen-dependent process, citric acid cycle, and the electron transport chain. So here's pyruvate that's generated during glycolysis. Now it's going to go to the mitochondria, but it's going to require a membrane transport. Because it's a molecule that does not go into the mitochondria by itself, it requires a membrane transport. So it goes to the transport. And once it's inside the mitochondria, it's converted into acetyl-CoA. This process releases 1 molecule of oxygen. Now, you must understand all of this process is going to occur twice because 2 molecules of pyruvate are generated for every glucose molecule. So for this process, in a way, we'll generate 2 carbon dioxide molecules, 1 from each pyruvate. And this process also generates, therefore, 2 NADH plus H pluses because there are 2 of them. This is an enzyme-mediated process. Now, from here, the acetyl-CoA, as you can see, is going to go to the Kreb cycle. Now, the purpose of the Kreb cycle is to harvest all the energy that's present in what remains of the pyruvate molecule. During the Kreb cycle, carbon dioxide will be released. Because there are 2 carbons left, 2 carbon dioxides will be released. And 3 NADH plus H pluses will be released. ATP will be generated, and FADH2, which is another electron carrier, is also generated. So these are electron carriers that are destined, as we have said, for the electron transport chain. That's where the real action is. So, and the fourth step is the electron transport chain, which produces some 32 to 36 ATPs. So now all the electron carriers will go into the electron transport chain, which is located in the inner mitochondrial membrane right over here, which is a series of proteins. So when we say electron transport chain, it basically means a series of proteins that are embedded in the membrane. They're transmembrane proteins, which transfer electron from one to another like hot potato. While they do that, they generate a hydrogen ion gradient, as we have previously suggested. And that hydrogen ion gradient is used to produce ATP by the famous ATP synthase enzyme, transmembrane protein. This process is called chemiosmosis, as we have suggested before. Chemiosmosis. This is the electron transfer chain. Now, the electrons are being donated by these electron carriers, the NADH, the FADH2, to the electron transport chain. As they go across the electron transport chain, their energy is being spent, so they have less and less energy. And finally, what are you going to do with them, right? You can't create a current, so to speak, right? So finally, those electrons are captured by oxygen and hydrogen, and water is produced. Okay, so the final electron acceptor, which is nearly always a popular biology test question, the final electron acceptor in the electron transport chain process in cellular respiration is oxygen. It is the final electron acceptor and is critical for this whole process. If the oxygen is not there, the electron transport chain does not work, and the whole of Kreb cycle and all this backup, and none of that will occur. So oxygen is very critical because it is the final electron acceptor. It keeps the system going and keeps the system in electrical neutrality. Okay. So this is the overview of the process. Now, so we talked about glycolysis. Now, pyruvate is taken by a transport protein, as we have suggested, in the mitochondrial membrane. And inside the mitochondria, it is converted into acetyl-CoA. So these two molecules of the pyruvate remain, and this carboxylic acid group over here is released as carbon dioxide. Remember, for every pyruvate, this is going to, for every glucose molecule, this is going to occur twice. Now, this is an enzyme catalyzed. Sometimes enzyme need help, and if enzyme need help, it calls a helper. So if enzyme need help, they call it helper. Their helpers are called coenzymes. Enzymes, coenzymes. And in this case, it's called coenzyme A, and coenzyme A is very important. Coenzyme A is basically grabs what remains of the pyruvate, and it's going to deliver this to the Kreb cycle. Basically, that's what it does. It's a mini taxi. So in this process generates one NADH plus H plus. Okay. Now, as you know, this NADH plus H, but basically it's an electron carrier. There are electrons there, right? There are two electrons there. So this is called reduction of NAD. To reduce a molecule is to give electrons to it, okay? So NAD plus is reduced so that it's produced NADH plus H plus. That's how you would say that. NAD plus is reduced to give NADH plus H plus, or just simply sometimes to save some breath, the precious oxygen is this as NADH. So here's the Kreb cycle. This is how it's going to look. Don't worry, we're going to go through it in a step-by-step process. So this is one mnemonic device I found to remember the different molecules of the Kreb cycle. So it starts with oxaloacetate. Now these sound very strange the first time you hear them. But after a while, they become reasonably comfortable. And at some point in my educational career, I remember I had to actually draw out the whole Kreb cycle with the molecular formulas and trace the carbons and everything. So you'll see this again and again. So even though they look intimidating in the beginning, after a while, they become like, old friends. Oxaloacetate, the first one. Citrate, second one. Isocitrate, third. Alpha-ketoglutarate is an alpha. The alpha-ketoglutarate. Succinyl-CoA is another CoA molecule. Succinate, fumarate, and malate. So oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate again. So this is a cycle which regenerates its starting substrate. If you want to remember this, one way to remember is our city is kept safe and sound from mice. Our city is kept safe and sound from mice. Or citrate is Kreb cycle starting. Citrate is Krebs starting substrate for making oxaloacetate. I like this one. Or cities kept safe and sound from mice. Oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and again oxaloacetate. So that's just a way of remembering some of these. So let's go to our Kreb cycle or citric acid cycle, one step at a time. So this is conversion of pyruvate to acetyl-CoA, releasing the carbon dioxide, as you can see. And acetyl-CoA reacts with oxaloacetate, 1, 2, 3, 4 compound. And this process requires water. And this generates citrate. Okay? Combination of oxaloacetate with acetyl-CoA generates citrate. Citrate changes to isocitrate. Okay? Basically, in this case, is moving this hydroxyl group from there to its neighboring carbon over here. Okay, so just kind of like shifting that hydroxyl. Isocitrate then is converted to alpha-ketoglutarate, which is over here. This process generates a high energy, high electron carrier, NADH, and 1 CO2 is released. Alpha-ketoglutarate is then converted into succinyl-CoA. This process requires the same, again, a coenzyme, CoA, and another CO2 is released during this process. Okay. Succinyl-CoA is then converted to succinate, and the coenzyme A is released so it can go back and help some more. This process generates a high energy compound GTP, which is like ATP, and this helps generate in one molecule of ATP like this. So GDP, okay, is converted to GTP, okay, and GTP can generate an ATP. Succinate is then converted to, sorry, I'm backwards, to fumarate. So this generates one molecule of FADH2. There are 2 electrons, and the 2 hydrogens are just tagging along because they're attracted to the electrons. So this is an electron carrier. And the fumarate, again, is converted to malate, or malate, and water. This requires water. And malate is, again, converted to oxaloacetate, generating, again, one NADH molecule. So look how much potential energy is over here in the form of electron carriers. So you can see Krebs cycle generates a lot of electron carriers, and these are all going to go to the electron transport chain. It also generates ATP over here. So this is a brief overview of the Krebs cycle. If you want to familiarize yourself with this, you actually might want to draw a Krebs cycle using the extra formulas of these one time, and then it might help you that way. From here, all the electron carriers are actually going to go and take their electrons and donate them to the electron transport chain, which is located in the inner mitochondrial membrane over here. These are the names of those proteins that are embedded in the membrane: NADH dehydrogenase, ubiquinone, succinate reductase, cytochrome BC1, cytochrome C, cytochrome oxidase, and this is the ATP synthase transmembrane protein. So these are the names of the electron transport chains. The NADH plus H plus is going to give its electrons over here, and the FADH2 is also going to come here and donates its electrons there. For every one NADH that goes to the electron transport chain, the math works out to it generates 3 ATPs, and for every one FADH2 that's donated donates its electrons to the electron transport chain, 2 ATPs are produced. So NADH carries a little bit more bang for the buck, so to speak. So this is how these are the names of the transmembrane proteins: NADH dehydrogenase, ubiquinone, cytochrome BC1, cytochrome C, cytochrome oxidase. And this is a cartoon representation. Now, as the electrons are being transferred, you see what happens is that the hydrogen ions are going to be moving across the membrane. So if I could draw this over here, I will use a different color over here. So electrons are be donated over here, and the electrons are going to go from here, transfer from one protein to another protein, from here to here, and as they're going by, they are going to take hydrogen ions over here across the membrane and generate a hydrogen ion gradient. In other words, the intermembrane space, the space between the two membranes, is going to be very rich in hydrogen ions, okay, like this, okay? And this process is going to generate lots of hydrogen ions over here. Now, this hydrogen ion gradient then is going to power this pump. They're going to go flow down here, and that is going to churn that pump, and this pump is going to generate ATP from its precursor ADP, adenosine diphosphate, and a phosphate molecule. So the purpose of the electron transport chain is to generate this hydrogen ion gradient. And finally, the final electron acceptor, as we have suggested, is oxygen. Oxygen plus hydrogen ions over here are going to take up the electron, and you're going to create a water molecule. Okay, that's the final electron. So that keeps the chain going so that more electrons can be donated. Okay, so that's how it is now. In this cartoon picture, you see in the end over here how water is being generated. Okay. Now, same thing, I think, over here in a little slow motion. Those black dots are electrons. And finally, water is created when those electrons are taken up, and the ATP synthase molecule. So here's what happens in a little more slower motion. So NADH plus H plus is going to give up its 2 electrons, and then there are going to be 2 hydrogens left, right? Hydrogen ions. And the electrons, carriers, are going to take those 2 hydrogen ions, they're going to transfer them across the membrane as they pass those electrons. FADH2 is going to come and donate its electron and hydrogen ions, and the same process is going to occur, coenzyme Q over here. And electrons, the same electrons are going to use to transfer those hydrogen ions across the membrane. And finally, here's the electrons. And those are going to be taken up by water. Like that. And then this hydrogen ion gradient, again, is used to create ATP. Like that. So this is the electron transport chain. Now, here's again, by the time you're done with this, you're going to really know this issue. Okay, so NADH gives us electrons. It's taken up by coenzyme Q. Now, the coenzyme Q is going to take those electrons and give it to this complex, number 3, protein complex. That's going to take some hydrogen ions across. That's going to take by cytochrome C. Gives us electrons over there to complex number 4. That's going to do the same thing. And finally, here it comes. Oxygen is going to come. It's going to produce that. So actually, H2O is produced. And then finally, ATP synthase is going to use that gradient to produce ATP. Some of the poisons, in fact, are the most deadly poisons known to men are those that destroy the electron transport chain. In fact, cyanide, as you've probably heard of the poison, blocks this transmembrane protein complex number 4, and it's a deadly poison for that. Now here's a count of all of the energy carriers, the electron carriers. So pyruvate being converted to acetyl-CoA. Okay. That generates some NADH, and acetyl-CoA is going to form citrate after combining with oxaloacetate. Citrate changes to isocitrate, and isocitrate is going to generate, and again, NADH2 releasing carbon dioxide, producing alpha-ketoglutarate. Alpha-ketoglutarate, again, it's going to give you one more NADH and carbon dioxide. And succinyl-CoA is going to produce GTP, which then makes ATP. The succinate reduces FADH2, gives us electrons to FADH to give FADH2, and fumarate turns into malate. And malate to oxaloacetate generates another NADH. So in this, going from pyruvate to the end of the Krebs cycle, you can see there are 3 carbon dioxides that are produced. Okay, so all of the carbon molecules that are in pyruvate, and therefore all the carbon molecules that are in the glucose, in a way, are released as CO2. So again, this process, glucose to pyruvate, occurs in the cytosol. The remainder of the process here occurs in the mitochondria, and the electron transport chain is located in the mitochondrial membrane. And that's it. That's the Krebs cycle. Inshallah, next time we shall talk about our next topic, Mendelian genetics. Until then, assalamu alaikum.